We all know topical medications, right? Creams, gels, but sometimes they have issues. They can be messy, separate over time, maybe deliver the drug too fast, or just not feel great to use. It's a real challenge. But what if there was a different way? A kind of semi-solid formulation that could fix these problems? Well, today we're doing a deep dive into something called bigels. It's a really fascinating, sort of newer approach to drug delivery through the skin. We're looking closely at a 2014 study by Singh and colleagues. They developed these bigels using guar gum and sesame oil. Pretty interesting stuff. So our mission here is to unpack this for you. What exactly are bigels? How are they different from, say, the gels or creams you might already know? And what did this research find about how stable they are, how they feel, and crucially, how well they deliver medicine safely? Let's get into it.
Okay, so maybe starting with the basics. Gels, most people have some idea. We have hydrogels, right? Water-based.
Right, water-based networks.
And organogels, which are oil-based. So what happens when you try to combine these? What makes a bigel? Yeah, that's what gets interesting. Bigels are basically a novel type of semi-solid formulation. You make them by carefully mixing a hydrogel and an organogel. But the really crucial thing, what sets them apart from your standard creams or emulsions, is they don't need surfactants or emulsifiers to stay stable.
No emulsifiers. How does that work then?
Yeah.
Usually oil and water, they don't mix.
Exactly. But here, the stability comes from the structure itself. One phase gets trapped inside the three-dimensional gel network of the other phase. It forms this unique, really fine dispersion. And that physical structure holds it all together. And that's a huge advantage, especially for skin applications. You avoid potential irritation or toxicity that sometimes comes with surfactants. It's just cleaner.
That really is a big deal. I mean, you look at ingredient lists on skincare stuff, and they're often packed with emulsifiers. So this sounds like a much simpler, maybe gentler approach.
Potentially, yes. Simpler in formulation, potentially gentler in application.
So given this unique surfactant-free setup, what did the researchers actually use to make these specific bigels? You mentioned guar gum and sesame oil.
That's right. They lean towards natural, well-known materials. For the water-based part, the hydrogel, they use guar gum. It's a natural polymer, comes from a plant, Cyanopsis tetragonaloba. It's already FDA approved, used everywhere, food, pharma, cosmetics as a thickener. Very safe.
Okay, makes sense. And the oil part, the organogel.
For that, they combined sorbitan monostearide, or SMS, with sesame oil. Now, SMS is technically a surfactant, but here it acts primarily as an organogelator.
Organogelator, so it helps gel the oil.
Exactly. It structures the sesame oil into a gel network. And sesame oil itself is a fantastic choice. It's been used for ages, for its own benefits, anti-inflammatory, antibacterial properties, even has antioxidants like sesamol that are good for the skin.
Wow. So the carrier itself has benefits.
Precisely. And then, to test the delivery, they used ciprofloxacin. It's a common broad-spectrum antibiotic used as the model drug here.
It's clever, isn't it? Not just a delivery vehicle, but potentially a system that works with the skin, using ingredients like sesame oil that have their own positive effects.
It's a more holistic approach to formulation design, absolutely.
Okay, so they made these bigels with different amounts of the hydrogel and the organogel.
Yeah.
What about stability? That's always critical for topicals. Did the ratio matter?
Oh, it mattered a lot. This was a really clear finding in the study. There was a direct link between stability and how much organogel was in the mix. The bigels with less organogel, they called them GG1, and UG2, well, they weren't very robust.
What happened to them?
They started to lose their structure, kind of flowed or separated after only 3 freeze-thaw cycles, or even just sitting around for about a month.
Not ideal.
Not at all. But the ones with higher organogel content, GG3 and GG4, those were the winners.
Yeah.
They stayed perfectly stable, held...
What did the extra organogel do?
It seems the sorbitan monostearide, the SMS, at higher concentrations created more extensive gel bonding within that oil phase, a stronger network, essentially. And they actually looked at this under a confocal microscope, too.
What did that show?
It showed that as you increase the organogel, the oil droplets dispersed within the hydrogel got smaller. They went from around 10 microns down to about 5 microns. And importantly, the droplet sizes were much more uniform, a narrower distribution.
Smaller and more uniform.
Right. This closer packing and uniformity really helps prevent the droplets from merging back together, which is what causes phase separation. It locks everything in place.
It means you can literally engineer the stability by tweaking that ratio. That gives formulators much more control over shelf life and product consistency.
Exactly. It makes the whole system much more reliable, both for manufacturers and for the person using it. No more separated creams.
Okay, so they're stable. But how do they feel? The texture, how easy it is to spread. That's huge for whether people will actually use a topical medicine.
Yes, the feel factor is crucial. And these bigels showed some really desirable properties for topical use. One key thing was shear thinning behavior,
Sheer thinning, huh?
meaning it gets thinner, less viscous when you apply force, like when you're spreading it on your skin. So it goes on easily, smoothly.
Okay.
But then when you stop rubbing, the viscosity increases again, so it stays put where you applied it, doesn't drip or run off.
That's exactly what you want, like a good lotion.
Precisely. They also showed a viscoelastic nature, which means they have properties of both liquids, flow, and solids, like elasticity or holding their shape. And again, the ones with more organogel were firmer, more robust, less likely to just relax or slump.
So more structure.
More structure, yes. And they were also more stable, thermally slightly higher melting points, which is good for storage and transport, means they hold up better in different temperatures.
These aren't just simple mixtures then. They have quite sophisticated mechanical properties. That shear thinning, the viscoelasticity, that really speaks to a well-designed formulation that considers the user experience.
Absolutely. It's about making it effective and pleasant to use, which boosts adherence to treatment.
Right. So stable, feel good. Now the big question, drug delivery. Did they actually release the ciprofloxacin effectively? And was it controlled? That's often the goal.
Yes, the drug release studies in vitro looked very promising. The bigels definitely provided a controlled release of the ciprofloxacin, which is often better than just dumping the drug all at once. You get sustained levels.
And did the composition matter here too, like the amount of organogel?
It did, and it was quite interesting. The release of the ciprofloxacin, which is lipophilic, meaning it prefers the oil phase, actually increased when there was less organogel.
So less oil meant faster release? That seems a bit counterintuitive.
It does at first glance, but think of it this way. With more organogel, the drug is more tightly held within that oil network. It acts like a reservoir, a depot.
Oh, okay.
So it partitions or moves out of the oil phase and into the water phase more slowly. The GG1 bigels, lowest oil, released about 40% in 12 hours. The GG4, highest oil, released only about 26%.
So you can tune the release rate by adjusting the oil content.
Exactly. More oil, slower. More sustained release. Less oil, a somewhat faster release. It gives you control. And importantly, the release followed zero-order kinetics.
Zero order. That means a constant rate, right? Independent of how much drug is left.
Precisely. It's like a steady drip, not a release that slows down as the concentration drops. That's often considered the gold standard for controlled delivery systems. Predictable and constant.
Wow, so tunable and zero-order release. That's impressive control. Okay, but what about safety? And did the drug still work after being put in the bigel?
Crucial questions. On the safety front, they tested for biocompatibility using human keratinocyte cells, skin cells. And the results were great. The bigel showed no significant toxicity, didn't harm the cells or change their shape, so they appear to be very gentle on skin cells.
That's excellent news. And the antibiotic activity?
Also positive. They tested the ciprofloxacin-loaded bigels against a bacterium, Bacillus subtilis, and found good antimicrobial efficiency. This confirms two things. One, the drug is stable and active within the bigel structure. And two, it's released effectively enough to kill the bacteria. The formulation isn't interfering with the drug's job.
Fantastic. So it works, it's safe, it's stable, it feels good, and you can control the release. That really covers all the bases.
It seems to tick a lot of boxes that formulators are looking for in an advanced topical delivery system.
So let's just recap this deep dive. We've looked at these guar gum and sesame oil bigels, and they really seem like a clever innovation. They're surfactant-free, which is a plus. They offer superior stability, especially with higher oil content.
Right, and those tunable mechanical properties, the shear thinning for easy spreading, the viscoelasticity.
And that controlled zero-order drug release, plus demonstrated biocompatibility and efficacy. It's quite a package.
It really highlights the potential of this bigel platform.
So thinking about the bigger picture, what does this mean for you, listening? Imagine your future topical treatments, antibiotics, maybe anti-inflammatories, who knows what else.
Yeah.
Imagine them being more stable, more effective because the release is controlled, but also just nicer to use, maybe even providing some skin benefits from ingredients like sesame oil.
Exactly. It moves beyond just delivering a drug to creating a better overall therapeutic experience.
And this controlled release concept, the stability,
Yeah.
it could work for so many different drugs, couldn't it? Not just ciprofloxacin.
Oh, absolutely. The potential applications are broad. Any situation where controlled topical delivery is needed could potentially benefit from this kind of system.
It really paves the way for smarter, maybe safer, definitely more user-friendly topical medicines. It makes you wonder, what other health challenges, maybe things like chronic wound healing or even advanced cosmetics, could be tackled using this Beigel approach? Where else could this combination of stability, control, and skin friendliness make a real difference?